For anyone interested in the findings from our paper, I've prepared this short video. The aim of this paper was to put together a set of simple recommendations for obtaining population heterozygosity estimates which would be suitable for comparisons across populations and across studies. We arrived at 3 recommendations. The first, to include monomorphic as well as polymorphic sites in estimates and thus not base estimates on SNP genotypes only. The second, to filter out sites with any missing genotypes. And the third, to analyze populations in independent runs. In arriving at the first and most important of these recommendations, we looked at how other studies using genomic sequence data had calculated heterozygosity. For studies analyzing individual genomes, heterozygosity is typically calculated as the number of heterozygous sites divided by the number of sites total, which we call autosomal heterozygosity. In this example, a single diploid individual is genotyped at 15 sites, of which 3 are heterozygous, giving observed heterozygosity of 0.2. If multiple genomes are analyzed, these estimates can simply be averaged. Here, sequencing 5 individuals or 10 individuals from the same population provides similar heterozygosity estimates that are based on the same proportion of heterozygous sites. However, when multiple individuals are sequenced, heterozygosity is often calculated from SNP genotypes directly. In this case, monomorphic sites will have been filtered from the dataset, but whether a site is called as monomorphic or polymorphic may depend on sample size. Rare alleles will be detected at a higher rate in larger samples, and if the rare allele is not detected, the site will be called as monomorphic and omitted from calculations. This biases the denominator of the calculation, leading to larger estimates of heterozygosity in smaller samples. Note that while this example here is demonstrating observed heterozygosity, also known as individual heterozygosity, these biases are also present for expected heterozygosity. Figure 1 from the paper shows how these biases play out across different sample sizes and filtering conditions. The subfigures with white backgrounds show estimates that consider monomorphic and polymorphic sites, which are consistent in magnitude. The subfigures with colored backgrounds show estimates using SNPs only, which are strongly biased by sample size. We also notice biases from including sites with missing genotypes, as seen here in Figure 2. The subfigures C, F, and I on the right-hand side show how observed and expected heterozygosities are very similar when sites with any missing data are excluded. But as missing data thresholds are relaxed, these estimates deviate. From this, we suggest that all sites with any missing genotypes should be excluded from analysis. A major advantage of including monomorphic sites in estimates is that the number of data points is greatly increased. 10,000 polymorphic sites may become millions or tens of millions of sites when not filtered by polymorphism. Thus, even after applying strict filtering for missing data, there will be plenty of sites remaining for accurate heterozygosity estimates. Our final recommendation is that when multiple populations are to be analyzed, that these should be analyzed in individual runs. This is simply to increase the number of retained sites. Subfigures C and D show that for 4 grasshopper populations, analyzing these individually retains 3.5 to 5 times as many sites, and about 5 times as many polymorphic sites. For autosomal heterozygosity, analyzing differentiated populations individually or together has no effect on estimates, as seen in subfigure B. However, subfigure A shows that this is not the case for SNP heterozygosity, which will be biased when differentiated populations are analyzed together.